Today we will be discussing our last set of notes for the semester, which is exciting. And our last set of notes is on DNA replication. So you're going to learn about different enzymes like DNA polymerase and what they do during the copying of DNA. We'll learn about the steps of replication, and then at the very end, we'll talk about the difference between prokaryotic cells and eukaryotic cells and how they replicate their DNA. So here's the structure of DNA that we know. Right, it's got a sugar phosphate backbone, the outside of the DNA molecule, nitrogen bases inside, and a double helix shape. Right, there's a little bit more structure to that, but that's the basics there. In its most basic form, DNA replication is pretty simple. It starts by having the old DNA get unwound. And so you see that here, the double helix starts to unwind into two separate strands. And then each strand grows its own new set of DNA over here. And so at the end, you'll end up with two. If we're talking about timing and when this occurs, you kind of already know that, right? If we know the cell cycle, DNA replication happens in the S phase of the cell cycle, or synthesis, right before the cell starts to divide. This occurs in the nucleus because that's where the DNA stays. And it happens because each new cell needs its own copy of the DNA. So I'm going to take you quick overview and then a little bit more detailed. We'll start with some basic parts, right? So the first step is that the DNA needs to be unzipped or unwound. There's a very cheesy joke that I apologize in advance for telling here, but it's really helpful. It goes, girl, I wish I was DNA helicase. That way I can unzip your genes. Terrible, I know, and it's not the greatest joke in the world. But it helps remind you what helicase does. Helicase is an enzyme that unzips your genes, or it unzips the DNA. It creates what's known as a replication fork. This replication fork, if we're talking about a eukaryotic organism, happen at a whole bunch of different places throughout the DNA. That way, it makes DNA replication go faster, and you can just stitch everything together at the end. But in general, if we're just looking at one part of the DNA, once DNA helicase unwinds the DNA, now you have two separate strands. One is called the leading strand, and the other is called the lagging strand. Another enzyme you need to know is called DNA polymerase. DNA polymerase is another enzyme. You can tell because it ends with ASE, like helicase. DNA polymerase has a few different jobs. Its main job is to add free-floating nucleotides to the single-stranded DNA to help build a double-stranded DNA. Right, so that's this molecule here and this molecule here. You see it goes along the DNA molecule. It adds these nucleotides there, and it also spot checks for errors, right? It's looking that A matches with T, C matches with G. And if it sees an error, it fixes the mistake before the whole DNA molecule gets glued back together. So that's DNA polymerase's job. So far we have helicase, which unwinds the DNA molecule, DNA polymerase, which adds free nucleotides. And the last one I'm going to say, which I don't know if I have a picture for, but I'll just tell you what it does. It's called ligase. Ligase is what is responsible for gluing everything together at the very end. Okay, so you'll hear this word called semi-conservative when we're describing DNA replication. And that's because you save some of the old DNA molecule when you are building the whole new DNA molecule. It works like this. Is it easier to have a page in front of you and make a copy of it, so you have two copies of it? Or is it easier to look at it and make a whole new copy by typing it up, right? Imagine I give you an essay to type. Would it be easier to just copy it in like a printer, copy it with your phone, make a copy that way? Or would it be easier to type the whole thing up again? That's the difference between DNA replication that would be semi-conservative, and then a DNA replication, which would be just making a whole new strand every time. In the semi-conservative model, what we have is that you're using the old DNA molecule as a template to build up the new DNA molecule. And since the base pairs always go together, if you know one half of the DNA, the other half should be pretty simple to fill out, right? If I had a base sequence of ACT, my new base sequence could be TGA, right? We're just matching the bases there. Now to add to the complication just a little bit. DNA replication is different on one side of the DNA molecule compared to the other side. One side is really easy. It's called the leading strand. The leading strand basically just has DNA replication happen continuously from the point of origin. DNA polymerase attaches to the DNA molecule, and it just continuously synthesizes the DNA in one direction. If you're looking at it here, the new nucleotides are being added from a 5' to 3' direction. That's a little bit more complicated than what happens on the lagging strand. On the lagging strand, synthesis happens discontinuously in little fragments. Those little fragments are called Okazaki fragments. You'll see the name in a second. But this happens because DNA polymerase can only work in a 5' to 3' direction. But what happens when the DNA molecule is being pulled apart, one side goes continuously in the 5' to 3' direction, while the other side is in a 3' to 5' direction, which means it has to wait for the DNA to fully separate out before it can keep replicating different pieces of it. All right, so here's a better picture where I think we can see it, right? Here's the leading strand, which the DNA is basically copied in that 5' to 3' direction. But this lagging strand, 5' starts up here in this like replication fork, and 3' is down here. And so DNA polymerase can only work in this like downward direction. And so what happens is that as more DNA is basically exposed and the replication fork opens up more, another fragment of the DNA will be copied. And it keeps being copied in these little fragments, little pieces over and over and over again down the lagging strand, making these Okazaki fragments. And then at the very end, you have a couple different enzymes that go through and basically connect all of these fragments. And that's part of what ligase's job is to do, is to connect all of these Okazaki fragments at the end. That's what we can see there in my slide is the ligase goes and connects all these Okazaki fragments at the very end. And so we have replication, which has become just a tiny bit more complicated by the fact that replication happens in two different directions. One side happens continuously, that's the leading strand, and the other side happens discontinuously in little fragments called the lagging strand. Last few things to discuss when bringing up replication. You have telomeres, which are little structures at the end of chromosomes. Basically, think of them as the protective caps. The enzyme telomerase adds short, repeated segments of DNA to the telomere as chromosomes are replicated to protect the end of the chromosome. So if there's any damage that happens at the end of the chromosomes while the DNA is dividing, it happens to little segments of the DNA that basically don't matter. Finally, if we're comparing prokaryotic replication versus eukaryotic replication, what happens in prokaryotic replication is because the DNA is circular, you're going to have two replication forks, and they're going to work continuously to build this new DNA until it's finally formed, and you have the two circular strands of DNA at the end, right? They work their way around. In a DNA molecule, one works this way, the other one works this way. We have the new DNA showing up here, while the old DNA gets replicated in the future. That's different from this eukaryotic DNA replication, where usually there's a replication fork at one point in the DNA strand, but there could be multiple replication forks along the long DNA strand here.